Okay, well, welcome back, everyone. So now we're going to head into part two of our talk about plants, where we're going to really focus on how plants sort of survive out in the wild, thinking about nutrients and water and thinking about their medium, right? Soil. So lots of, I think, more interesting stuff than the first part of our plants lecture. We'll really first sort of focus on the nitty gritty of plants, thinking about plant biology and function and structure, and then plant groups. But now we're going to sort of, again, I think a much more interesting topic, dive into sort of how plants do their thing. So, the first aspect of thinking about this is the biological interface that plants have with the environment. Now, everything interacts with the air in some shape or form, but plants are unique in that half their, well, depending on the plant, part of their body is indirect contact with the soil. Right? That beautiful stuff that you've probably called dirt, but is super important for plants who are growing where we walk on the Earth's crust. Now, soil is the highly weathered outer layer of Earth's crust comprised of sand, rocks, clay, silt, humus, air, and other forms of minerals, as well as organic matter. Now, all 92 naturally occurring elements occur in the Earth's crust, and most are found in the form of what we call inorganic compounds called minerals. Soil as a piece, as it sort of says, also contains microorganisms. And as we discussed with, like, say, the mycorrhizal fungi, these microorganisms are potentially very important to plants. Now, we're going to break the soil down into different layers, but we'll start with the most important, which we call the topsoil, which is where most of the roots are found. Plants do most of their stuff in topsoil. Now, topsoil is a mixture of minerals and particle size that vary, include as well as living organisms and dead organic material, which we call humus. Now, it is characterized by relative amounts of sand, silt, and clay. So the ratio of sand to silt to clay really dictates the type of soil you have. Now, interestingly enough, depending on where you go in the world, you'll have more sand versus silt versus clay. So, for instance, here in Boston, we have quite a bit of silt and clay versus, say the American Southwest, which has a lot of sand with very little silt and clay. So, again, depending on where you are, the ratio of sand and silt to clay in the topsoil does vary. Now, the soil composition also determines the degree of water and nutrients that bind to soil particles. So sandy soils hold on to very little water, silty and clay soils hold on to very much more water. So let's look at our profile. We have the area where the plants are living, which we typically call the leaf litter and where the plant light lives. Then we have that beautiful topsoil, and that's basically where all the action is happening. That's where microorganisms are, worms are, burrowing creatures, the plant roots, all the good stuff is in that topsoil. Then we have the subsoil, which goes deeper down, which is comprised of minerals that have been leaching from rocks and those that have leached from above. And then we have the bedrock, which is weathered material, very rocky, not really suitable for plant life. So again, most of plant life is happening in this topsoil, with some of it happening in the subsoil. But again, most of it is in the topsoil. Now, the availability of minerals does vary. Now, minerals, as you guys, as a sort of a concept, right, probably recognize dissolve in water. Now, minerals, in terms of how they're available, are only available to plants and to other organisms if they are dissolved in water. And so, if they're dissolved in water, they're readily available for uptake by plant roots and other organisms present. Now, um, the really interesting thing about this is that most soil particles are negatively charged. That's just a function of soil. It's mostly negatively charged. So as a result, biologically important positively charged ions such as calcium and potassium, they like to bind to soil particles. However, when water is present, water will liberate them from soil particles, allowing them to be uptaken via channels in the plant's roots or the root hairs, allowing the plant to pull it in and transport calcium or potassium or whatever it is from the soil. Again, that water pocket around soil into the plant roots. Now, we like to think of soil as being a hard thing, right? We walk on we feel like it's like a hard surface, but the reality is when we actually dive into soil as a thing, soil is anything but that. Depending on the soil where you are, some soils have immense amount of pores. And what do I mean by pores? Well, it's pretty nicely illustrated in this figure. we have lots of pockets in between particles of soils. And that's what we mean by pores in soil. Now, these pores in the soil, as it's illustrated in these three figures, can be a combination of air pockets or pockets of water. But the reality is soil is not just a hard surface. There's dynamism to it, right? There's holes, there's pockets of water, there's pockets of air, and so on and so forth. But things that are held in small pores or open pores, but not in the air, are readily available to plants as a whole. Now, topsoil, as I've mentioned, is the weathered component of soil. Now, topsoil loss is pretty common. As soils lose more and more water, their capacity to stay together is significantly decreased. So very, very dry soils, as I'm sure you guys have probably seen in like maybe like a Disney movie like Aladdin, right? Very, very dry soils, like there was an Aladdin in the Sahara Desert, well, they easily get blown away by the wind. And that's the true, not just for say the Sahara where things are very dry, but that's the case in all aspects of soil. The drier they are, the more likely our soil is to blow away. And so one of the sort of the best case scenarios we've seen of this here in the United States is actually the Dust Bowl in the 1930s. For those that have never heard of the Dust Bowl, due to extended drought and poor farming techniques led to massive erosion of topsoil and eventually the loss of sort of the togetherness of topsoil, leading to massive dust storms pulling off tons and tons of topsoil, creating these huge dust storms and creating this large amount of famine in the United States, which we collectively call the Dust Bowl. So now, how do we prevent erosion of soils? Well, the reality is plants do a really good job of preventing erosion. Now, as we sort of just kind of look here, when a plant is growing in soil, its roots are going out. So plants actually help to hold soil together. But from an agricultural standpoint, to prevent soil erosion, we have to do a couple different things, including intercropping, conservation tillage, and as well as no-till um, growing of crops. And all these things are effective at preventing soil erosion, but the reality is we actually just don't do that. So scientists actually predict we're heading towards another dust bowl in the near future, unfortunately. But again, we can prevent soil erosion by doing these things. Now, soils come in different flavors, and I actually mean that. You can actually taste the difference between acidic soils and basic soils and neutral soils by actually how they taste. I know that sounds gross because you'd be eating dirt, but acidic soils taste tangy. Um, pH, neutral soils don't really have a taste to them, and very basic soils have kind of like a soapy taste to them. Why? Because it's just a reflection of the pH. So many soils are acidic, and the pH of soil does affect the release of minerals from weathered rock. So, for instance, at very low pHs, aluminum, which is a toxic compound to plants and is also the third most abundant element in the crust, readily leaches into the soil and into pores, thus stunting the growth of plants. So it's kind of a bummer thing. But that being said, low pH isn't a bad thing. Most plants actually grow best at slightly acidic pHs, about a 6.5 in that ballpark. And so, put that in perspective, about 26% of the world's arable soil is actually considerably more acidic than optimal. So, we grow lots of food in places that are already very acidic. So, just as a note. Now, as we mentioned, there's also saline soils out there, which is due to the accumulation of salt ions, typically sodium and chlorine, after water potential. They alter water potential in soil. This overall leads to a net loss of water and turgor pressure in plants, as we discussed. Now, saline soils are far more common in dry areas where salts are introduced through irrigation. So we see this in the Great Salt Flats of Utah. Very, very saline soils because it's also very, very dry. Now, one of the other common things we see in soils is this idea of desertification. Now, we see this here in Iraq, not Iraq, Iraq, in the 90s, where things got so dry, it led to soils, which were normal, in this case, where boats were, just becoming completely deserts because of so much loss of water. So deserts pose a huge sort of thing. So those are our different soil stuffs. I didn't talk about alkaline soils. Alkaline soils aren't very common. Most of the world is slightly acidic to neutral pH, just as a note. So we don't really focus on alkaline soils too often. Now, plant nutrition is an important part of their interface with the soil. Now, photosynthesis, as we all know, is the major source of plant nutrition. They capture atmospheric CO2, they fix it into sugar using energy from the light. Makes sense, right? That's what photosynthesis is. Now, plants also need a number of nutrients as well. Now, they need lots of macronutrients. There are 9 in particular. COHN, so carbon, oxygen, hydrogen, nitrogen. They also need potassium, calcium, magnesium, phosphorus, and sulfur. Then there are 7 micronutrients, which are used in smaller amounts: chlorine, iron, manganese, zinc, boron, copper, and molybdenum. And a deficiency in any one of these can have some pretty severe effects on plant growth. So I've given you a handy dandy table here, different elements that are important, the principal form which is absorbed in the plants, and then the percentage of dry weight. So you can see carbon and oxygen come together to give us about 88% of plant biomass, with oxygen being 6%, nitrogen being 1 to 4, and so on and so forth. So really handy dandy table for you to take a peek at when you have a few minutes for macronutrients, and then followed up by micronutrients. But we can physically see mineral deficiencies in our plants. So if you've ever tried to grow a plant, you can actually diagnose how poorly it's doing by the color of it and whether or not you see what's called chlorosis. So you see here, here, and here, a change in the color of the plant from the nice standard green to a different color is chlorosis. And so different types of chlorosis give you different effects. So for instance, this type of chlorosis is a phosphorus deficiency. This type is nitrogen deficiency. And you can really diagnose how poorly your plants are doing depending on the color of their leaves in terms of if they have a deficiency in minerals. Now, plants need ammonia, NH3, and nitrate, or I should say, or nitrate, to build amino acids, right? Nitrogen is a key part of DNA and amino acids, the building blocks of everything. However, plants and nitrogen, well, actually, let me take this back. Nitrogen as a whole is relatively rare on the planet. And I say nitrogen as in bioavailable nitrogen, nitrogen such as ammonia or nitrate. So it is relatively rare. Now, there are organisms that take atmospheric nitrogen, N2, and can actually fix it into ammonia. This process is called nitrogen fixation. But plants lack that capability, as do most organisms on the planet. So what plants have done is they've evolved symbiotic relationships between plants and bacteria called nitrogen fixing bacteria. Now, we can actually look at these bacteria. The most common genus of bacteria that do this are the rhizobium. Now, these bacteria require oxygen and carbohydrates to support their metabolic pathways. So they're heterotrophic like we are. So the plant gives the rhizobia oxygen and food. In return, the plant also houses the bacteria in these nodules. And we commonly see these in plants like legumes, like beans. And inside these nodules, the bacteria actually convert that atmospheric nitrogen, this N2, into ammonia for the plants. And there's a really interesting way this develops. And so I just like to talk about it briefly because I find it really fascinating. We have the epidermal cells and the cortex cells in the plant root. The plants secrete these organic compounds called flavonoids, which recruit these rhizobium bacteria. In return, once the rhizobia get there, they secrete these nod factors that change the biology of the plant. Which cause them to build up on the plant, causing the plant to form essentially a loop around the bacteria, allowing the bacteria to form what's called an infection thread, invade the plant, then start anchoring itself inside the plant, developing what we call bacteroids, which are basically inert or not motile bacteria that fix nitrogen inside the plants. And then we have the full maturation of the nodule where we have all these bacteroids or nitrogen fixing rhizobium inside the plant. And again, nice predictable fashion. And again, commonly used by members of the legume family, not just beans, but also like alfalfa, peanuts, and so on and so forth. So lots of things in the legume family have evolved this symbiosis. Now, the other one, which we've already talked about, is the mycorrhizal associations, which are symbiotic associations with fungi that we know we found in about 90% of vascular plants. Now, as we discussed, these substantially expand the surface area available for nutrient uptake, and they enhance the uptake of phosphorus and other micronutrients. Now, those that don't have these types of symbioses, whether it's mycorrhizae or rhizobium, have actually evolved the carnivorous life cycle, which leads us to the really fun carnivorous plants. Now, these typically grow in very acidic soils that lack nitrogen. Now, we actually have these in Massachusetts. We have lots of bogs in Massachusetts. And inside these bogs, we have numerous carnivorous plants. And so how do these work? Well, the first kind is actually a pitcher plant. And you'll notice a pitcher plant just kind of looks like a pitcher of water. It's got an opening. And inside this opening is just like this cavern. And inside this cavern, there's all this slippery hairs, and then a pool of digestive liquids. So what these pitcher plants do, because they need nitrogen, and there's really no nitrogen in the soil, they trap insects in their little digestive fluids, digesting the insects, leaving all the dead insect parts in there for bacteria that live in there to eat. And then they suck up all the nitrogen into their plants, thus being carnivorous plants to get their nitrogen. So I think the pitcher plants are wild. We have these in Massachusetts. You should try to find them if you have spare time one day. And then the one that you're probably more familiar with, which is the Venus flytrap. Now, you guys probably recognize the Venus flytrap. It's basically just a giant mouth. Um, it has sensory glands inside. It's essentially its mouth, where if an insect lands on it, it tickles it, causing the mouth to close. Then once the mouth is closed, it secretes digestive enzymes, dissolving the insect, releasing um, releasing all the nitrogen from the insect. So again, a way to get nitrogen out of our organism. And then the final one is the sundews. In this case, it's from the genus Drosera. They have these little sticky ends. They have these sticky mucilage secretions, traps the insects, then they release digestive enzymes, getting the nitrogen out of the insect's body. So rhizobium is great, nitrogen is great, but predatory plants such as the ones we've seen, including water wheels, trap insects, essentially digest them, getting the nitrogen out of them. And as you would imagine, because of their life cycle, they're actually all related to them phylogenetically. So just as a note, really cool way of getting nitrogen. Now, you can get your nitrogen from bacteria, you can get it from the soil, you can get it from insects, or you could be a parasite. which is I think probably one of the more interesting. But parasitic plants are interesting. They can be photosynthetic or non-photosynthetic. And as of now, we know about 3,000 species of plants that do this. And what they do is they rely on other plants to capture nutrients for them. So they are parasites. They take they take energy from plants. They take nitrogen from plants. So, for instance, the Indian pipe is actually a non-photosynthetic one. It essentially tacks onto other plants. In this case, it's a tree, sapping nutrients away from the tree, um, being a parasite from the tree. So taking nitrogen, taking phosphorus, taking carbon away from the tree. They're still plants, but they're parasitic plants. And so these ones, for instance, you can find these here in Massachusetts. If you go to pristine forests, where there's not a lot of pollution, you can find these. So that's for Indian pipes. So those are all the ways we get nitrogen, but carbon is a really important aspect of plant biology, and the ratio of carbon and nitrogen inside of a plant body is important. Now, the ratio of this tells you how healthy the plant is. So when you look at the ratio of carbon to nitrogen, the higher the ratio, typically the worse off the plant is doing, i.e. it has less nitrogen relative to carbon. The lower this ratio, typically the better this plant is doing. Now, the IPCC, or the Intergovernmental Panel on Climate Change, has concluded, as have every other scientist on the planet, that CO2 is at its highest levels in the last 20 million years, which correlates with increased activities from humans. So we're thinking about climate change here. Now, increased CO2 levels alter that CNN ratio of plant, which again is important for the health of the planet, plants, as well as the thing that eats the plants. Now, why is that the case? Well, plants take carbon dioxide, they stuff it into the Calvin cycle, fixing that CO2 into sugar. As you probably remember, photosynthesis is primarily catalyzed by this enzyme Rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase, which catalyzes the first step. Now, what's interesting about this, it can actually bind to carbon dioxide or oxygen. So if CO2 binds to Rubisco, a 3-carbon sugar is made that can be then used to make glucose and sucrose. If oxygen binds, photorespiration occurs, which results in neither nutrients or energy storage. So too much CO2 typically is a better thing. Too little CO2, not enough good thing. But this is what photorespiration looks like. So again, plants can use photosynthesis to make sugar or to help them respire. And again, all we're looking at here is the Calvin cycle, which we covered in Bio 1. But again, producing no sugar, but respiring in photosynthesis. Now, there are also other types of photosynthesis out there. There's C3 and C4 photosynthesis. So C3 photosynthesis occurs in our mesophyll cells. This is the standard photosynthesis that we think of. Now, C4 photosynthesis actually uses an extra pathway to shuttle carbon deep within the plant leaf. Now, what happens is this reduces photorespiration as well as water loss by limiting the Calvin cycle, so the part that fixes carbon, to cells surrounding the vascular tissue where oxygen levels are in fact low. Also in areas where water loss is significantly lower. So what ends up happening here, CO2 comes in our C3 plants here. It comes in, it's fixed in the Calvin cycle, sucrose comes out. And again, it happens in these mesophyll cells. Now, in our C4 plants, CO2 comes into the mesophyll cells. There's an enzymatic pathway, fixes it into a different form, It fixes into a slight acid. Then that acid is converted back to CO2 inside the bundle sheath cells where it goes into the Calvin cycle. And again, we see the mesophyll cells here, the epithelial cells here, and then we can see the bundle sheath cells here. This kind of looks like a face, like a screening face. That's what I've always thought. Now, in C3 plants, as CO2 increases, the Calvin cycle becomes more efficient, which in theory, with climate change, should lead to increased photosynthesis and growth. However, if plants have less nitrogen and minerals per unit mass, i.e., the ratio of carbon to nitrogen is higher, this results in lower nutritional value for herbivores, and more plant matter must be eaten to obtain the same amount of nutrients, and plants are doing more photosynthesis than they should be. So that's plant nutrition. Now let's talk about how plants reproduce. So how plants produce the next generation. Now, in particular, we're thinking about angiosperms here. Now, angiosperms, as we discussed, they're the major evolutionary innovation with the production of flowers and fruits. And plants go through several developmental changes leading to reproductive maturity by adding structures to existing ones with their meristems. So let's look at the life cycle of an angiosperm here. We'll start with the zygote, the two end. We have embryonic development into the seed, and then eventually into the fruit, which then leads to the dispersal of the seeds, leading to the sporophyte generation, i.e., an immature plant, leading to the maturity of the plant, leading to a fully mature plant that produces flowers. Then the flowers produce their gametes. So remember, they produce the pollen in the anthers and their eggs in the ovule. And then we have fertilization occurs, which starts this process over and over again. So that's the life cycle of our angiosperms. But let's talk about how flower production is regulated. So once plants are competent enough to reproduce, i.e., they're old enough, several factors, including light, temperature, and in working together, promote and inhibit internal signals, which in turn determines whether a flower is produced or not. Now, this undergoes what we call a phase change, and sometimes it's really subtle, sometimes it's very obvious. So, for instance, when a flower is getting ready, it'll go from a juvenile, where it looks like this, to an adult, which looks like this, and then in a combination of factors for temperature and light, which produce either floral promoters, i.e., starting flowering, or floral inhibitors, which prevent flowering from occurring. Now, this phase change, again, is internal developmental changes that allow plants to attain competence to respond to external or internal signals. Now, this might be morphologically obvious or very subtle. Again, pretty morphologically obvious here. Other times it's very, very subtle. So in this case, we have the oak tree. Now, the lower branches in the oak tree actually retain their leaves in the winter because those lower branches are actually initiated by that allow it to cling to walls. So you can, it's sort of hard to see in this picture because there's isn't quite showable, but ivy will make little adventitious roots that allow it to cling to walls. But after a phase change, the mature ivy, as we see here on the right, actually lacks the ability to produce those adventitious roots. So it's kind of an interesting thing with a phase change. Now, there are factors that affect the flowering here. So, for instance, in Arabidopsis, the delay of flowering is actually controlled by the EMF gene or the embryonic flower gene. And this helps prevent early flowering. Now, we can have mutants that flower immediately. Just as a note, there's always mutants for any gene out there. So, there are mutants that flower immediately. But flowering is the default state, and many mechanisms have evolved to delay flowering as a thing. Now, you can imagine flowering is energetically expensive, and you'd absolutely be right about that. So, delaying your flowering or having missed time flowering is not a particularly good thing for you. But to induce flowering or that juvenile to adult transition can be induced actually by overexpressing a flowering gene that's relatively conserved across most plants, which we call leafy or LFY. Now, leafy was actually cloned in Arabidopsis and replaced with a viral promoter that actually results in a high levels of leafy expression. Now, transcription, I should say. And overexpression of leafy in aspen causes flowering to occur weeks instead of years, which is kind of crazy, right? So, for instance, normally speaking, aspen trees, which are relatively large trees, they grow for several years before producing flowers. However, by genetically altering aspen, we can cause the overexpression of the leafy gene, causing rapid flowering in aspen, and again, in this transgenic aspen. aspen, not aspirin, sorry. But yeah, pretty cool. Phase change controlled by genetic machinery. Now, flower production has four genetically regulated pathways that have been identified thus far. So the first is the light-dependent pathway, the temperature-dependent pathway, the gibberellin-dependent pathway, and the autonomous pathway. And plants can rely on one pathway primarily, but all four pathways can be present in a single plant, just as a note. Now, as we discussed, plants' flowers have pretty distinct structure, and floral organs were actually thought to evolve from leaves. And so a complete flower has four whorls. We call them calyx, the corolla, the androecium, and the gymnosium. And an incomplete flower lacks one or more of those whorls. So let's define some flower morphology, and then we'll look at some pictures. So the first one is the calyx, which consists of flattened sepals, the corolla, which consists of petals, the androecium, which is the collective term for all the stamens, the male structures of the flower. Then the gynosium, which is the collective term for all carpels or the female parts of the flower. So let's refresh ourselves. We have the sepal, we have the receptacle, we have the ovule here, then we have the carpel, we have the stigma, style, ovary, and ovule. Then we have the petals, we have the stamen as well. So this is our complete thing. And here's the handy dandy part. All the stamen parts are the androecium, all the carpels are the gymnosium, all the petals are the corolla, and all the sepals are the calyx. So that's our complete flower structure. Now, there are some pretty distinct trends in floral specialization. The two major trends are floral patterns have grouped together and floral parts. Whereas advanced flowers are bilaterally symmetrical. Radially symmetrical would just be symmetrical around a circle. Bilaterally symmetrical is on an axis, right? So if I divide this flower right down the middle, it's the same on the left and right side. Bilateral symmetry is something we'll see when we talk about animals in our next lecture, just as a note. Now, gamete production, as we've talked about, is the alternation of generations, where we have the diploid sporophyte leading to the haploid gametophyte. But in our flowering plants, that gametophyte generation is very small, and it's completely enclosed within the tissues of the parental gametophyte, sporophyte, I'm saying. So remember, when we think about this, just as a note, the sporophyte generation is here and here, but it's completely closed in the sporophyte generation. Um, Now, the reproductive organs of our angiosperms, as we know, they're specialized structures of the flower, and those reproductive organs of angiosperms are different from those of animals in two distinct ways. So both the male and the female structures usually occur together on the same individual, whereas animals, there's a male and a female, right? Um, and then the other idea is that reproductive structures are not permanent parts of an adult individual, right? The penis of a human doesn't go away, right? It's not a temporary thing, but for angiosperms, it really is. So let's just sort of just refresh what gametophyte formation looks like. We have the anthers here producing our pollen, and we have the ovules here producing the eggs. We have that double fertilization event that occurs here, going from the sporophyte to the gametophyte generation. But remember, the gametophyte generation is still housed within the sporophyte generation for both our male and female parts. But then we have the mitosis of both of them producing megaspores or microspores. Then we have fertilization leading to the overall production um, of a fertilized egg, leading to the development of all the parts. Now, how does pollen form? Well, the anthers contain four microsporangia, which produce microspore mother cells. Now, these mother cells produce those microspores as we've seen here, right? Um, I'm sorry, the mother cell here producing the microspores we see here. Sorry, looking at the wrong thing. Then the microspore develops by mitosis into pollen, and then the generative cell in the pollen grain will further divide to form two sperm cells. So back to where we are, so we have the microspore mother cell, which is produced in the pollen sac, leads us to microspores, which leads us to the tube cell, the pollen grains, which have the generative cell and the tube cell nucleus. The tube cell nucleus again going to produce the sperm. Now we can look at some 3D structure of pollen, and if you're like me, you might be allergic to pollen. I'm very allergic to pollen. I get really bad allergies in the spring. But pollen has some really cool 3D structures, and each species of plant produces very different structures of pollen. So for instance, on the left here, we have the eastern lily, Lilium candidum. Now its pollen tube actually emerges from a pollen grain through the groove on the furrow that occurs on the side of the grain. So we're talking about these areas. That's where the pollen is going to emerge for. Now in the plants of the sunflower here on the right, three pores are actually hidden among what we call the ornamentation of the pollen grain, and the pollen tube may grow out through one or more of them. So we're talking about things that are, hidden here, here, and here. Pollen may grow out of one of them. So again, pollen structure varies depending on what species of plant you have. Now, within each ovule, there is a diploid megaspore mother cell that undergoes meiosis to produce four haploid megaspores. Now, usually, as we discussed, only one megaspore survives, and this enlarges and undergoes repeated mitotic divisions to produce 8 haploid nuclei. These are then enclosed within a 7-celled embryo sac. And again, this is what we've seen thus far. But again, this is what our mature embryo sac looks like. We have 2 antipodals, 2 polar nuclei, the egg, and then synergids. And this is our overall structure of our embryos. Now, for pollination to occur, pollen is placed on the stigma, and then there's 2 types of pollination. So the stigma remaining to the end part of the female reproductive part. Once pollen gets there, it can travel down through the anthers, to the style, down to fertilize the ovule. Now there's 2 types of pollination. There's self-pollination for when a flower's own pollen pollinates its own stigma. So it's basically like making a baby with yourself. Or cross-pollination from when pollen from one flower of one species pollinates the other species, or when pollen from one flower pollinates the egg of another flower on the same plant. So just as a note, that's the sort of the 2 distinct groups of pollination that we have. Very diverse group of organisms. We like to think of them as being bees, but it really encompasses so many groups of insects as well as many animals. And there's a huge amount of coevolution between flower morphology and the pollinators that pollinate those flowers. Now, early seeded plants, such as those of our gymnosperms, were wind pollinated. However, insect pollinated angiosperms, the most common pollinators are those by the bees. So we'll talk about the bees because the bees are certainly important. And if you wanted to know why it's called the birds and the bees, well, this is where it comes from. Now, bees typically visit yellow or blue flowers, and many of the bees, many which have stripes or lines on that indicate the location of the nectaries. So pollination works because flowers provide a sweet, delicious treat, nectar, that bees or other organisms can eat, which entices them to come to the flower, picking up the pollen and bringing it to another flower. Now, bees are really interesting. This is how we see a normal flower here. In case this is a yellow flower, it's Ludwigia privania, or the Peruvian primose. This is what we see. This is what a bee sees. Why is this the case? Well, the outer sections of the petals, actually, interestingly enough, reflect both yellow and ultraviolet light, while the inner sections only reflect yellow light. Therefore, they appear darker to the bee, thus drawing the bee in to where the pollen is, where the nectar is. Thus, flower pollination can be successful. So bees are the most common, but there are many other types, including butterflies, moths, there are beetles, flies, and so on and so forth. But in terms of other major pollinators, birds are pretty dang common. Birds such as the hummingbird here like to come to flowers. Why is that? Well, flowers produce pollen and nectar. I'm sorry, they produce nectar, nice sugary treat, which is good for everyone. Now, flowers that are pollinated by birds are typically red in color. So they are visual to birds, but they're generally ignored by insects just due to the visual patterns of um, bird's eyes versus insect's eyes. Now, there are other types of pollinators for animals, rodents, bats, birds, insects, many other types out there. But typically speaking, one flower typically has its own set of pollinators. Say if a hummingbird goes for flowers XYZ, it will ignore flowers ABC, for instance. So pollinators can be pretty specific. Now, some angiosperms, like gymnosperms, are wind pollinated, which, as we discussed, was a characterization of early seeded plants, such as our gymnosperms. Now, these flowers, typically speaking, are small, green, and odorless. Common way flowers attract individuals for pollination is by sweet smells. One of the reasons we like the way flowers smell is because of the scents they use to lure in pollinators. Um, And so what this typically looks like is we have the female flowers on top and the male flowers on the bottom. The wind comes in, picks up the pollen from the male flowers, bringing it over to the pollen of the female flowers on another plant or the same plant, thus allowing fertilization to occur. That's one example. Another example here is we see the large yellow dangling anthers, again, picked up by the wind. Again, then the female flowers here aren't pictured, but again, the large male ones picked up, carried to female flowers. And this is actually pretty common on grasses, just as a note. So now self-pollination is a thing. Now outcrossing or cross pollination is much more advantageous for plants and eukaryotes as a whole, right? Sexual reproduction is awesome for more genetic diversity, and more genetic diversity is a good thing. However, self-pollination is pretty common. Now, there are two basic reasons for the frequency of self-pollination. The first is self-pollination is favored in environments where pollinators are scarce, so deserts in particularly, and the offspring are actually more uniform and probably better adapted to their environment if self-pollination occurs. So generally speaking, genetic diversity is good, but sometimes when you live in really harsh environments, low genetic diversity is a good thing. Now, outcrossing is promoted by several evolutionary strategies, including the separation of stamens and pistils in space. So, in particular, dioecious plants that produce only flowers, ovules, or pollen. So, remember, these are plants that are either fully male or fully female. Or we have monoecious plants that produce male and female flowers on the same plant, but they mature at different times. In this process, what we call dichogamy. Then we also have this idea of self-incompatibility that actually prevents self-fertilization. So we have these three mechanisms that actually promote outcrossing, preventing incrossing. And again, we can look at our dichogamy here. So this is our monoecious plants where we have a male plant and a female plant, and they have this sort of pollen transfer between them. And again, but it moves through physical space to have that pollen transfer. Um, allowing this to occur. Now, there's also self-incompatibility, which also increases outcrossing. Now, the pollen and stigma do recognize each other as being genetically related, and actually the pollen tube growth is actually blocked. So a plant just basically prevents itself from being self-fertilized, which is, I think, from a evolutionary perspective, is pretty dang good, right? I argue that's probably better to have more outcrossing as a whole. Now, it's controlled by alleles at what we call the S locus. Now, there are two types of self-incompatibility. The first is what we call gametophytic, which is gametophytic self-incompatibility, which depends on the haploid S locus of the pollen and the diploid S locus of the stigma, so they're not compatible. Or we have what we call this sporophytic self-incompatibility, which is in the alleles in the stigma will match either the pollen parents' S alleles and the haploid pollen will not germinate. And so this is where we have here so this is where it's determined by the haploid pollen genotype, and this is determined by the genotype of the diploid pollen. Again, not too important. This is not too important thing to remember, but just know there are incompatibilities that arise between pollen in egg that also prevents self-fertilization from occurring. Now, we also talked about this process of double fertilization, which again only occurs in angiosperms. That requires two sperm cells. Remember, double fertilization results in the fertilization of the egg and the formation of the endosperm that nourishes the embryo. So, just a refresher what this looks like. Pollen comes in, it has the tube cell and the generative cell. It comes into the stigma down the style, making its way to the ovary, making its way to interact with the uh, uh, embryo sac. There we go. Now, what ends up happening is the sperm all comes all the way down. The sperm comes in. One sperm fertilizes the polar bodies. One fertilizes the egg, leading to the endosperm nucleus, which is 3-end, and the zygote nucleus, which is 2-end. But again, this is our process of double fertilization, producing the egg, which develops into the plant, and the endosperm nucleus, which develops obviously into the endosperm. Now, once the egg is fertilized, we have embryonic development. Now, This growing pollen tube, as we mentioned, grows into the angiosperm embryo and releases two sperms, fertilizing those two structures we just meant, which leads us to our first zygotic division. Now, the first zygotic division is asymmetrical, resulting in cells with two different fates. So the first one is the small cell, which divides repeatedly to form a ball of cells, which will then form the embryo. Then the large cell divides repeatedly, forming an elongated structure called the suspensor, which transports nutrients to the embryo. Now, the root shoot axis also forms at this time via cells near the suspensor become the root, and cells at the other end become the shoot. Now, let's look at the early cell divisions here. So we have the polar nuclei, we have the sperm kind of coming in, doing its thing, producing the endosperm and the diploid zygote. Now, this leads to the first cellular division, producing our suspensor here in dark blue. There's a basal cell here, and then the endosperm here. Then we have further mitotic divisions producing the globular proembryo. And again, we still have the endosperm and the basal cell and the suspensor here. Then we have the cotyledon start to develop over time. And again, nothing has much changed outside of the development of the cotyledon. And then we have the cotyledons fully developed, giving it sort of like a peace sign shape. Then we have the development of a couple different things. So we have the ground meristem starting to develop. Then we have the procambrium starting to develop, the protoderm being down here. Then we have the root apex, which is also called the radicle, developing here. And then we have the shoot apical meristem down in here. And then once things are finally done, we still have the cotyledons. We have the root apical, the shoot apical meristem, the root apical meristem, and then we have the hypocaudal here, fully giving us the sort of the full standard structure that we've been looking at up to this point in time. Now, the zygote actually develops sort of interesting. So we're going to talk about a really interesting process in fucus. So it's a brown algae. Now, the development of the zygote in fucus actually is a result of asymmetrical cell divisions. What ends up happening, we have a bulge developing on one side of the embryo where cellular division occurs, resulting in a smaller cell that develops into a rhizoid that anchors the algae, and then the larger cell that actually develops into the thallus or the main algal body. So actually, let's look at this. We have the zygote. Light and gravity influence it. It forms a bulb, which leads to the rhizoid cell. And as it develops, if we have a full-on thallus, full-on rhizoid, giving rise to this rhizoid, which again is this anchor, which fully develops into, again, this anchoring point for algae. And then the thallus fully develops into the form roots. How I'm going to form shoot? Well, in plants, that three-dimensional shape and form arise from regulating the amount and pattern of cellular divisions. Now, the vertical axis, i.e. the root shoot axis, becomes established very, very early on, as we discussed. And the same is actually true for the establishment of the radial axis, i.e. the inner-out axis. And then the first cells themselves physically divide, producing a solid wall of cells. Now, apical meristems, which are, again, the site of cell growth, right? That's where the plants are growing either vertically or horizontally. They establish the root shoot axis in the globular stage from which the three basic tissue systems arise: dermal, ground, and vascular, as we discussed in the previous lecture. Now, these tissues themselves are actually organized radially around that root shoot axis. Now, primary memory stems themselves will actually differentiate while the plant embryo is still in this globular shave, thus no cell movements are involved. However, the outer proderm will develop into dermal tissue that protects the plant, and the ground meristem develops into ground tissue that stores food and water, and the inner procambrium develops into vascular tissue that transports water and nutrients. Now, this leads us to this topic of morphogenesis. Now, morphogenesis kind of sort of spells out what it is, but we'll walk through exactly what it is. Now, the globular stage itself gives rise to heart-shaped embryos with bulges called cotyledons, as we've seen thus far. There's two in eudicots and one in monocots. Just as a note, we haven't mentioned that before. We've always been looking at eudicot embryos, but again, there is a differentiation between eudicots and monocots here. Now, these bulges are then produced by embryonic cells and not by the shoot apical meristem, which we call morphogenesis, which results from changes in the planes and the rates of cellular division. Now, as a plant is dividing, there are critical developmental events. So during embryogenesis, angiosperms undergo three critical events. First being the development of a food supply, the second being the development of a seed coat, and the third being the development of fruit surrounding the seed. Now, there is considerable variation in the endosperm, and it varies pretty heavily between plants. Now, in coconuts, this includes the liquid milk. So, in case you like coconuts, you are eating endosperm, and it also includes the milk of coconuts, which is so tasty. In corn, it's actually solid. It actually expands with heat to form the white edible part of popcorn. Just as a note, if you're a popcorn enthusiast like me, you like popcorn because you like endosperm. And then finally, in peas and beans, it's actually used up during embryogenesis, and the nutrients are stored in thick, fleshy cotyledons. So if you like beans, you're eating very thick, fleshy cotyledons. And again, we can see it here. This is our corn. So you have the cotyledon here, the endosperm here, and the whole embryo structure here, giving us this standard corn structure we end up seeing. Now for legumes, in this case, the red kidney bean, you have the very large cotyledon, the embryo, but again, no endosperm at all in our beans. So interesting dichotomy between the function of the endosperm in these two distinct groups of organisms. Now, seeds, as we've discussed, are an important function of both gymnosperms and angiosperms, but in many angiosperms, the development of the embryo is arrested soon after the meristems and the cotyledons differentiated, leading the seeds basically becoming dormant. Now, we have the teguments develop into a very essentially impermeable seed coat, and this encloses the seed with its dormant embryo and stored food. Now, you might ask yourself, how does a seed know when to germinate? Well, the emergence of the first root, which we call the radicle, um, is actually just piercing the seed coat. And germination itself, as we've discussed, cannot take place until oxygen and water reach the embryo. Now, some seeds have this process called stratification that requires periods of time at low temperatures before germination. So many agriculturally viable plants, um, like wheat, for instance, require a very cold temperature for it to actually the seeds to actually germinate. But seeds store food, and germination and early seedling growth does require the metabolism of energy reserves inside the seed. And this is principally as starch. Now, we have what's called the suctellum, which is our kernels of cereal grains, where the single cotyledon is actually modified into the structure. So it's a monocot, which transfers nutrients from the endosperm to the embryo. Now, there are also hormonal effects here that, in response to the absorption of water by a seed, the embryo produces what we call gibberellic acid, and these signals what we call the aileron, which is the outer layer of the endosperm, to produce amylase, which digests starch. So levels of abscisic acid, which is another plant hormone. I should just make mention gibberellic acid is a plant hormone, just that's a note. Levels of another hormone, abscisic acid, which actually inhibits starch breakdown, also are reduced during this time when the seed absorbs water. But again, we can look at this interplay here. So we have the embryo here, we have the cotyledon or the suctellum, which again transfers things between the endosperm and the nucleus, the embryo here. Now, when water comes in, it sort of stimulates the production of gibberellic acid, which again activates the production of an enzyme called amylase, which breaks down starch into sugar. So, from the molecular level, that's how things are regulated with hormones. But ultimately, this leads to once a seed is in a happy state, excuse me, it leads to germination, where we have the seed coat, the early root system is being established in what we call the primary roots. You can also see the secondary roots starting to pop up here as well. But essentially, the seed essentially folds out of the ground in sort of like this shape as you're seeing here, eventually becoming first um, uh, very sort of the very first early vertical stem, which leads us to the structure which we call a plumule, sorry, which is the early leaves. You can also see the cotyledon here as well. Now the plumule develop into essentially the first leaves. You can also see the cotyledon here starting to wither and die. And as the plant gets bigger and bigger and bigger, the cotyledons eventually start to disappear. So this is one way we see it. Germination can also occur. This is beans, just as a note. We can also see this in corn, where we have the radicle comes down, forming the early roots, that primary root. Then we have a stem pop up, leading to the first leaves, and then developing roots, and then the development of the first plants. And we can also see those specialized roots we see in corn here, right? Uh, so that's our that's how germination works. Again, just going from the seed structure that we know and probably don't love, but we certainly know it now, into these fully mature plants. Now, asexual reproduction is not uncommon, but it does produce genetically identical individuals because only mitosis occurs. And this is pretty common in harsh environments, um, such as deserts and salty areas and so on and so forth. Now, this does lead to this process called apomixis, which is the asexual development of a diploid embryo in the ovule. Um, kind of an interesting thing. So that's one way, asexual reproduction. We can also have what's called vegetative reproduction, where new plants are cloned from parts of adults. So this actually comes in many forms. We've talked about runners previously, but we can also see a few others. Coming here, including rhizomes, suckers, and adventitious plantlets. There are some plants where you can actually cut like stems off and put it in water, and it'll actually form a brand new plant from a stem. So you can have this process of vegetative reproduction. Now, I think sort of one of the sort of questions that might come up in your brain is how long do plants live? Well, plants have a variable amount of times. Now, woody plants such as oaks and pines and so on and so forth have pretty extensive secondary growth. So, so going fatter essentially, which leads them to having very longer lifespans. So, most oaks that you see out, say in the Boston area, are sometimes hundreds of years old. And so, the wider a tree is, the more secondary growth has occurred. The taller a tree is, the more primary growth has occurred. But the more growth you have, the older the plant is going to be. Now, some of the oldest plants on the planet include our bristlecone pines, which can live upwards of 4,000 years. Um, but depending on the life cycle of their plants, whether it's a woody plant or otherwise, plants can be annual, biennial, or perennial. And I'm sure you guys have probably heard these before if you've ever bought plants at the store or something like that. But let's talk about these plants. What does annual, biennial, or perennial mean? Now, annual plants grow, flower, and form fruits and seeds within a single growing seasons. Then they die when the process is complete. So if you want a plant that comes back every year, you don't want an annual. Biennial plants have life cycles that take 2 years to complete, and they store energy for 1 year, and then they flower in the second year. And then we have the perennial plants, which continuously grow year after year after year, which can either be herbaceous, so you can think of things like a rose bush, or woody, such as oak tree. And we can just see examples here. We have desert annuals here, and then we have perennials here of our redwood trees. So just as a note. And that's going to be the end of our talk about plants. I'm sure many of you are very excited about that, as plants I know aren't the most exciting thing. But next lecture, we're going to pick up talking about organismal biology, talking about animals, the evolution of animals, and all the cool things about animals as a whole. So I hope you guys learned a little bit of something. And please let me know if you have any questions. Otherwise, take care.